Thank you, Dr. Chao. Okay, I'm continue the second lecture about the diagnostic applications of chromosomal disorders. Today's outline include first is that I would discuss the recent development from traditional cytogenetics to cytogenomics, and then introduce copy number variation and the method of detecting it, and then followed by examples for detecting genomic structural variation using various techniques. As you may know, in 1990, we have the, just a minute, I have something. It covers here. How do you do this? Oh, okay. Okay. Thank you. So in 1990, we started the fluorescent in situ hybridization and molecular, that's the start of a molecular cytogenetics. And then in the same year, also discovered genomic imprinting and UPD, understood as important mechanisms of human genetic diseases. In 1992, they developed a technique called comparative genome hybridization. Usually is to study solid tumor study. Basically is the patient DNA and control DNA hybridized together, mixed together and co-hybridized to the metaphase chromosome to see if there's any changes. And usually this is a very good, is very commonly used in solid tumor studies. In 1996, there was a 24-color fluorescent in situ hybridization, meaning is also known as a sky or spectral karyotyping. And that is that they're using multicolor and then manipulate it in such a way that each homologous chromosome have its own color. So for example, when you have a tumor and there were some multiple chromosome exchanges, and by the color, you know the origin of the chromosome. And so that was very popular also in cancer study. Until 2001, chromosome microarray became to study. But it's not until 2004, we are the first laboratory using the chromosome microarray analysis, in short as CMA, into the clinical laboratory. And then subsequently, multiple other laboratories are also using different technique, different sort of microarray to use it in the clinical laboratory. And by multiple studies, and they review, I think they review about 30-some studies and declare that the array CGH is the first test replacing G-banded chromosomes. And then 2011, that the next generation sequencing to the clinic by way of a whole exome sequence, that is the WES, usually short-read technology, also implemented. We are also the first implemented this WES test. And then because of on the research field, they have the undiagnosed network that NIH funded for multiple center, and we are one of the centers studied all the patients who had WES tests, but it's normal. And so they start studying, and then they're using that there's a team to study this involving the clinical geneticists, biochemical geneticists, molecular geneticists, and cytogeneticists as team to evaluate these. And finally, based on from that experience, then we also published in New England Journal by the name of Peng Fei Liu. They reanalyzed the West. And at the same time in CUHK, Elvis Dong and Richard Choi developed the low-pass sequencing to identify structural variation. In 2019, Baylor also implement the whole genome sequencing. And as you know, that also the same year, Science published, the magazine Science published the completion of the entire genome from telomere to telomere for each chromosome. It's long read using the long read technology, but this is still in the research arena. And by the time you guys come to study, probably will be a new era to study the using long read instead of a short read technology. So to remind everybody about the genome with the high resolution. Traditionally, you see the chromosome on the one end is G-banded chromosome on your right, and this chromosome these are blood chromosome, or you could do other chromosome, but the blood chromosome had a high resolution, but the highest is the resolution is about 4 megabase. In contrast, when we use the FISH fluorescent in situ hybridization, the resolution is about 40 to 250 kb. And you can see that the chromosome using the fluorescent probe to study the presence or absence of the gene. You know. And usually that is about well, all these we call for the FISH, like the 40 to 250 kb, if the loss or gain, this we call copy number variation, CNV. And then the molecular study usually is studying the resolution, 1 base pair to 1,000 base pair. And why we study CNV? Because CNV, the mutation of CNV is 10 to the minus 4th, 10 to the minus 6 to 10 to the minus 4th, where the mutation rate for the single nucleotide polymorphism is around -8. So it's 1,000-fold more common in CNV than SNP. So that's why the CMA became very because CMA is between the single nucleotide and the G-banded. So this is a very good intermediate studying. And today we're focused on this area. As I mentioned last week, we studied chromosomes, and this week we study CMV. So what are CMV? CMV is copy number variation and intermediate scale genetic change defined as segments greater than 1 kb or 1,000 base pair in length, but typically less than 5 megabase. Because after 5 megabase or more, then you could usually, if 10 megabase, you probably should be able to see it in chromosome. So the CMV, very common in human genome. So, there are a lot of the, there are 2 kinds of CMV. One kind is because it's very common in the human genome, so they are normal. because it consists of 25, almost 30% of the reference genome. And so we have to learn how to distinguish what is a normal CNV versus disease-causing CNV. And roughly the distinction is that normal individual, there's a database called their database to collect all these common cause of normal variations, identified in about 35% of the human genome. In general, they are smaller in size, contain fewer genes, highly variable region, such as near the centromere region, and the segmental duplication, and often inherited. In contrast, the human disease-causing genes, one of the most common cause of human disease. The diagnostic yield is 10 to 20% in developmental delay, intellectual disability, autistic spectrum disorders, and birth defects. In general, they are larger in size, contain more genes, and located in unique region of the genome, and often de novo. So as you can see that on your left is a high resolution chromosome 1. And if you see that there was a red bracket that showing the 4 megabase region on your right, that if there's a 1 megabase deletion, you won't be able to see it by a high resolution G banding. So therefore, the CMA became popular because they could detect these micro deletion or micro duplication. In this incidence is 4 megabase region of microarray data showing 1 megabase deletion encompassing 70 oligos. So at Baylor, I'm going to speak more about the Baylor experience, that we chose to using adjuvant array. And the reason we're using adjuvant array because they had an extensive library of probes, and we could choose the probes and use the probe that place them into a disease-causing region rather than just randomly distribute probes. We use so they had 4 different fab formats. One is 1 million, there's also 400K, and there was also 180K and then 60K. Right now, the clinical array we use is 400K. So in one slide, you could do 2 patients. And the clinical utility of microarray is detect copy number variation in human genome, deletion and versus duplication, and structural rearrangements, and also enable the identification of critical gene that had altered copy number and may be responsible for the development and progression of a particular disease. And there are 2 types of array. One is the by Agilent, is called microarray comparative genome hybridization, used to detect copy number changes in the human genome. So we call that as a CMA, chromosome microarray analysis. There's another kind, is SNP array, single nucleotide polymorphism microarray for uniparental disomy pair of chromosome and diseases causing for autosomal recessive genes. Nowadays, that the adjuvant array also provides some SNP into the array. So, but that the array is only 50K SNPs. So usually we do it as a screen. The SNP arrays now produced by Agilent, no, by Alpha-Matrix and Illumina. And my focus today will be on the Agilent array. So the process of process of a CMA is that you can collect sample from CVS, amnio, blood, placenta, and other tissues. After DNA extraction, the step one is to label the DNA, label the patient DNA and the control DNA. And one is green and one is red. And you mix them together. And then you hybridize to array of in this array of DNA and probe in the slide, whose location is known. So in principle, that if the patient DNA is more than the patient, okay, if the patient DNA and reference DNA mixed together, nothing gained and nothing lost, that will be yellow. That means that patient has 2 copies and control has 2 copies, and the ratio will be 1:1. If the patient DNA is greater than the control DNA, that means that would be patient had 3 copies versus 2 copies of the control. The ratio will be 1.5 to 1. And that we call a gain. We can't using deletion and duplication. Using this, we can only say copy number gain and copy number loss. Until you finish using fish to confirm they are deleted, then you could call that deletion, or gain, that's a duplication. And if the patient DNA is less than the reference DNA, then it will be red, right? So that means the patient DNA had 1 copy and control DNA had 2. The ratio will be 0.2. The ratio would be 0.5 versus 1. So therefore, is a lot copy number loss as indicated in the bottom graph. So after after the hybridization onto the array, and the slide will be put in the scanner, and the scanner will read these red, green, yellow spots. And then using computer, we have a in-house software that typically here is representing a typical output of CMA using our in-house software detecting copy number loss or gain. And this incidence, so you can see that here the log ratios, if nothing lost and nothing gained is here. And here's our chromosome 1, 2, 3, 4, all the way to X and Y. And then if nothing lost, nothing gained will be in here. However, you see that on chromosome 17, there's a copy number loss. So it's showing in red. So the copy number variation is as I mentioned last week, that the human chromosome composition is in pairs, right? And so it's 2 copies. So it's 2 copies each. And when the genome fragment show deletion or duplication after FISH, it will cause CMV. For example, like this copy number, you only have the normal one, you have 2 copies, right? So you lose 1 copy, then it's a copy number lost, and you gain a copy is copy number gain. So how to interpret the copy number variation detected by microarray? So if it's normal, if it's normal, the log ratio, if the fluorescence signal side 3 of the sample to be tested in the probe area is 550, the fluorescence signal is side 5 of the normal control is 520. So the log ratio is log 2 ratio 1 equal to 0. So like just remember I said the yellow and the green, their copy number, no change, is all in here at the 0. And this yellow dot is usually we use it as a control. So we'll place some polymorphic clone in there to indicate that this as an internal control. So if anything, gain will be on this way, on this side, and the loss will be on this side. So if the fluorescent if the log ratio of fluorescent signal side 3 of the sample to be tested is 250, such as in here, versus the control 520, then the log ratio is -1. Here is the 1. And then when you open up this chromosome and find out that the deletion is concentrated into this NF1 gene, and NF1 gene is associated with neurofibromatosis type 1. And you will see patients with this neurofibroma across the part of the body, the whole body. And you know this patient is had associated with this patient had neurofibromatosis type 1. And this is this scenario because they lose 1. It's a copy number loss here. In contrast, if the fluorescence signal side 3 of the sample to be tested in the probe area 790 versus the control 520, then you got the log ratio of 2 will be 0.5. So you will see this gain here. And that means that in your mind that you mean that that's 3 copy gain. It could be more than that, but generally you usually lose 1 copy. So the example of copy number loss, you would have if it's a heterozygous deletion, then you would see that it's only half of the you only get half of it, half of the normal control. And so this is the mutation, or this is, as I mentioned earlier, that's the neurofibromatosis. So the log ratio is -1. Now, if there are homozygous deletion, that means the fluorescent intensity of the sample to be tested is 0. The log ratio will be more indefinite, usually is more than -2. And that might also indicate that, this patient may be like cystic fibrosis. You have 2 copy deletion in order to get the autosomal recessive disease. So here's the example of a homozygous deletion 2 copy number loss. You could see this is this is on chromosome 8, and there's a big deletion below 1.5 minus 1.5. And it turned out to be that this is a homozygous deletion of VPS 130B, 13B. And when you open up the chromosome, you open up the chromosome 8. So here is the chromosome 8 and all the probes. And you could see that this is going all the way down to -6. And so when we look at the graph, we know that this the location of this gene, the location of this deletion is from the location of this copy number loss is and this is the location, the maximum and the minimum and the maximum location. So the loss was 51 kb, and then it's on this VPS 13B, and you could see that it's a copy number loss, and the total is 46 probes, and that is -4. Medium range is -4.2. So in your mind, that is both copy loss. And Cohen syndrome is associated with this loss of this gene is associated with Cohen syndrome. And here's the child with associated with Cohen syndrome. It's 5.5-year-old boy seen for autism, and he had myopia at 6 months of age, and has a depth perception issue, hypersensitive to sound and sensory issue related to his autism, central hypotonia, frequent infection, documented neutropenia, global developmental delay, and diagnosis with autism before the age of 2. But they are repetitive behavior, including hand flapping and twirling, does not have any words. And this is because we find out that he had this copy number loss, autosomal copy number loss on both chromosomes, inherited in the autosomal recessive manner and associated with Cohen syndrome, because the gene is that VS13B is completely, after we do the fish, then both of the gene is completely deleted. And that's how we make the diagnosis. Example of copy number gain. So normally, the normal, you had two copies of but sometimes the patient had 3 copies. So it would be one chromosome is normal, the other one had 2 copies. So the log ratio is 1.5, and the log2 ratio would be 0.5. But there's another one is homozygous deletion or homozygous duplication or triplication. Then you will have your 2 scenario. You could be 1 chromosome normal, and then you have 3 copies on this side, or you could have both of chromosomes are duplicated. An example of this one is amniotic fluid sample in 21 weeks, 3 days for abnormal ultrasound. And the patient also had a genesis of corpus callosum. And as you can see in the plot, that the the plot that is all the things are normal except on chromosome 17. It turns out there was 1.3 megabase gain of 70p12 on the CMT1A critical region containing the PMP22 gene. And this caused Charcot-Marie tooth, and which is an adult onset peripheral neuropathy. So these usually are inherited. So we study the parents. It turns out the mothers also carry this gain. And there's a terminology is that, so usually even if the fetus have this CMT1A, is usually don't exhibit until adulthood. This is adult disease onset peripheral neuropathy. And usually, for example, usually the mother behave very normal, and because of the pregnancy and find out this, and then we studied the parent, the mother has it, and we call this as incidental finding. And that's another terminology, and that we will in counseling you run into this situation. So here, just as I mentioned, why the significance is well known. This is adult onset disorder. Such finding can be incidentally detected. And so even the mother, if she's young mother, she probably might not exhibit the symptom yet. So this is the picture that I got from the Google to find out these CMT affected, that were affected their legs and arms and hands and legs. And the example of triplication, this is Peng Fei Lu who wrote this paper. There's the PMID if you're interested. PMID, if you're interested to reading the article, you just write down this number and you go to PubMed, and then you'll get the article. So they study a family that this is the father and this is the mother, and the mother has the CMT. And you could see the fish, usually the green is a control, and then you could see that, or usually it's a centromere, so that's thicker. And then the probe that we want to test is so you could see there's two, and there's two here. So that's this one. When it's duplicated in this scenario, the normal one would be this one, a green and red. If it's duplicated, you could see that there's the green is a control, and then this is duplicated. And then when study, they produce a twin, and the twin, the male twin turned out to be triplicated. And you could see that this is the normal chromosome, and this is the triplicate one. So here you could see 3 dots. And then the daughter is duplicated. Again, see this normal 17, and it's duplicated. And when we examine their phenotype, and you could see that you could see that the 3 copies, the hands and legs are affected. But when it's 3 copies, you could see that it's more severe. So this is example. I introduce you another concept called dosage sensitive gene. Because later on we find out that this PMP22 gene in the CMT1A region. When this gene is mutated, they also could associate with the CMT1A. So that's why we know this gene is called disease-causing gene. When it's one copy, meaning that one copy is deleted, so the other copy it's only one copy. The patient result in HNPP. In short, actually, is hereditary neuropathy with liability to pressure pulses. When it's doubled sensitivity, when it's duplicated, then you would have CMT1A. If it's triplo sensitivity, then you have a very severe T1A. So that's what we call dosage sensitive gene, because this gene, different dosage would have manifest different phenotype. Now, a little bit about the SNP array, because the SNP array, they could detect copy number changes. But because it's using SNP, single nucleotide polymorphism, we could detect it as known as absence of heterozygosity, which I will show you in the next few slides. So the important thing to remember, detecting of absence of heterozygosity, we could detect uniparental disomy, such as Angelman syndrome, Prader-Willi syndrome, and other UPD. Both can be detected postnatally and prenatally. And by calculating the megabase of the AOH, if they're going to be more than 400 megabase, you could detect the region. So it's known as detection of regions of identity by descent. That will contribute to autosomal recessive disorder. And if it's more such as a first cousin mating, but if it's like more than 400 kb in common, that you might suggest there's incest, such as the sexual abuse of a young female at home. For example, it's the father-daughter mating, then you produce about 400, lots of more than half of the genome is have this UPD. So this is how the SNP array can detect this. How this is determined? So in the healthy individuals, the SNP1, you had the paternal and maternal. You have this ATGT, notice A, and this is all same, right? Ooh. Ooh. So we call homozygous. SNP2 and 3, the father and this is a C and this is a G, this is a T and this is a A. So we call that heterozygous. And then in SNP4, again, is A and A. So this is, so we call that homozygous. These are healthy individuals. So here is the display of SNP allelic difference pattern. For example, you had the father's AA, and so you read it here, AA. So here's the AA, AB, and BB. Or you could do this BB, AB, and AA. And then you could see, oh, And then here, then you will have BB, BB, and then AA, BB, and AB. So if it's single allele, then you'll be a copy number loss. If it's triple allele, there will be copy number gain. So the way it reads that is that this is the array plot, the CMA plot. So it's normal. And then in here is the SNP plot, allele frequency plot. So this is the AA, this is the AB, and this is the BB. So a normal individual, you would have AA, AB, and BB. But if it's lost heterozygosity, then you would have you don't have AB, either AA or BB. If it's a gain, then you would involving AA, AAB, ABB, and BBB. Then you have 4 lanes. So when it's a deletion, the B allele frequency can be either 0 or 1 because it's either A or B, meaning that 0 and 1 are. And here is a different one that you see this. Only here is a loss of heterozygosity. Here is normal. So how you distinguish uniparental disomy versus deletion? Uniparental disomy meaning that there are 2 copies, right? So in the log ratio, it's normal. And then when it comes to B allele frequency plot, you could see that either AA or BB. So that means that both copies are from one single parent. Where in a deletion, you could see the plot, the log ratio plot is a deletion, and then therefore is absence of heterozygosity. So you have to, when you determine whether it's a UPD or deletion, you have to look at both the log ratio plot and B allele frequency plot. And then when it's duplicated, then you have this scenario as indicated in the circle, that you have the gain on the log ratio plot, and then B allele frequency, you would have BBB, ABB, AAB, AAA. So you have 4 lanes. So how we're using this? This is one good example on this is chromosome 15. And then you could see there's absence of heterozygosity in the proximal 15. Here, this is a centromere and a chromosome 15. And the AOH is only happening here. And this is because this is an echelon SNP array. The probe is not as heavy as the Illumina and Alpha matrix array. And then you could see that AOH. So when we study Prader-Willi syndrome, there are 4 scenarios can cause Prader-Willi syndrome. Majority of them, this is the normal, one from father, one from mother, and they are normal. Whereas if the deletion is in the paternal side, then 65 to 75% of them, if it's deleted the paternal allele, then you have Prader-Willi syndrome. Another 20-30% if both chromosomes are from the mother, from maternal side. So that means it's absence of the paternal chromosome, then also exhibit Prader-Willi syndrome. And then about 2 to 5% is an imprinting disorder. That means that there was some mutation in the imprinted gene in this region. So now I finished talking about the log ratio, the CMA, and then also the SNP pre-allele frequency. So now we're going into introduce you some of prenatal examples or different examples to illustrate what we learned. Here is a sample is a direct CVS, 13 weeks and 3 days. The indication is advanced maternal age, abnormal ultrasound, that means a nuchal thickening, and karyotype performed at another laboratory and show normal chromosome. So this is probably 10 years ago. So the lab couldn't figure out how to interpret, so they send us because it's normal, right? So they sent to our lab for array, and this array, when the array when we run the array, then you could see that everything is normal except for chromosome 4. And this chromosome 4, when you open up there, it turned out to be 8.7 mega base loss, because we also do fish confirm it. At then 4Q21.23 at Q22.1. And when you open the chromosome 4, here's the whole plot, and then you could see this. So you could see that the location, and then the maximum, a minimum start and maximum start, because we never know exactly the breakpoint, because these probes are descriptive. That you know the last one that's gain and the last one is loss. The last one is normal and the next one is loss, and in between. So we don't know the breakpoint. That's why we have the minimum and maximum. The minimum is meaning that the loss, the first oligo that it indicate loss, and the last oligo indicate loss. That's the minimum start and the maximum stop, the minimum stop and the maximum stop the same way. So that's why we have two different location. So in here is at least is 8.66 8.67 megabase deletion, and it could be potentially as big as 8.7. And here is all the gene that is involved. And then there's 417 probes involved in this region. So the average medium is close to is minus 0.94, close to -1. And as I mentioned earlier, the CMV, when we report the CMV minimum and maximum size. This one is from here to here is the minimum size. Here to here is the maximum size, because we don't know from here to here where the exactly breakpoint deletion. So in our report, we'll indicate the minimum and the maximum size and the CMV location. This is the location, and how many genes within the reported region that they would print it out, all these disease genes. And so just review the copy number loss, because uh, the normal is 0, right? And then heterozygous deletion is -1. So one is normal and the other one is minus, is loss, copy number loss. So we fish, we subsequent, well, to using the, we using fluorescent in situ hybridization uh, to confirm the deletion, or confirm the copy number lost. And then now we know it's a deletion because this is a centromere of chromosome 4. And this is the probe that we use in that region, and that's deleted. So that's why we know that's deleted. But because it's 8.7, because it's 8.7 deletion, megabase deletion, it should be. So we thought that we should be able to see it. And as you can see that even this is not a very high resolution bending, you actually can see it. This is the normal 4, and then you have this region, and then this region is deleted. And same as here, this band and this band is deleted. So you could also check it out from the UCS browser by typing the coordinates, the coordinate in here. Then you could see that this portion is deleted, and then you click out on the UCSC browser that you could you could click on ClinGen, or you click on Decipher, and those are in each red or blue. Red is deletion, blue is duplication, and so you could want to see if there's any patient similar to what this patient represent. And because this is 8.7 megabase deletion and involving multiple genes, then as a cytogeneticist, you think that this could be reported, this could be exist in the literature. Turns out that I was right, that we find a couple, actually several reports, but this the report shows a chromosome more better than the other ones. Then in this article shows that they have 2 patients with de novo proximal interstitial deletion of the long arm of chromosome 4. In one of the deletion resulted in monosomy 4q 4q21.3 and q23, which is another. They actually find 9 cases this region, and these patients are in short is associated with central nervous system overgrowth, facial anomaly, hypotonia, developmental delay. And as you can see, you can see that this band and here this band is missing. So when you counsel the patient, you cannot say the patient is going to be having this. You would say, when you counsel the patient, you say, patient with this deletion, patient with this will be associated with these abnormalities. Because that was a newborn, is unborn child, you don't know how that developed, but you have seen other patients with this deletion associated with these abnormalities. Another clinical case is 20 weeks, 3 days gestational age. They have abnormal ultrasound, hydrocephalus, sacrococcygeal dysplasia, cystic hygroma, and bronchoatresia, and pulmonary cysts. And so we run it on an array, and we find out that there's a copy number loss, copy number gain, and copy number loss, as indicated in the circle of this chromosome, entire chromosome 6. Usually, when you see that copy number gain and copy number loss involving in the same chromosome, usually is involving inversion. And I'm going to show you how that we determine that. So then you would have the copy number gain, as you see here's the copy number gain, and then on the 6p term, sorry about this, and then copy number loss on the bottom, and then you see that it's 6q term. First one is 6p25 is almost close to the 6p term, and the low one is 6q25.3 to q27, and then there's how you write your report with this nomenclature. And so when we go back to study patients with this deletion, it turns out that we also wrote a paper on this, that patient with the terminal deletion would have 6q would represent with structural brain abnormality, including a genesis of corpus callosum and hydrocephalus. Remember, the patient we have is a hydrocephalus, so it's sort of consistent. Periventricular nodular heterotopia and cerebellar malformation. A fetus with a similar size of deletion has been documented as presenting prenatally with hydrocephalus and lumbar hemiinvertebra. So 6q deletion, currently this gain and loss of an opposite arm of the same chromosome are suggested for unbalanced recombinant chromosome that across the abnormal that arose from a abnormal transmission of apparent balanced pericentric inversion. Therefore, parental chromosome or FISH analysis is recommended to determine if this finding is de novo or familial. And when we see the chromosome, actually, for those trained, that we will see that the chromosomes inverted. And as you see, they indicated the arrow, they had clearly one more band, 1, 2, 3. Ooh. Keep going. 1, 2, 3, and here it's just 2. So you have this a little bluff that this one didn't have. So it turns out that this inversion find in the father, and it turns out that this portion and this portion is inverted. Apologize for that. So I just want to illustrate to you that this portion and this portion is inverted. So when it's if this is inverted during the meiosis, the pairing, they cannot pair regularly they couldn't do the homologous pairing. Instead, they would have to this loop form inversion loop results. So let me see if I could. This is too sensitive. Let me see if so because it's this is a normal 8. This is a normal. This is a normal 8. So you have the blue one is a normal 8. You have 1, 2, 3, 4, 5, 6, 7, 8, right? And the inversion one, you would see 1, 8 is intact, but 2, 3, 4, 5, 6 is inverted. So when it pair, and if there's a crossing over, you could see that this 8 go all the way back here and crossing over to here and form this kind of scenario, where the other one would be this one and go all this way back to this one. So you would have 2 possibilities. If crossing over occur between the inversion breakpoint in meiosis 1, duplication and deficiency chromosome will be formed. So this is the duplication, because 8 and 8. Deletion, and then deletion, because in this one, you miss the 1, right? You have the 8 and 8, and you're missing 1. And here you have 1 and 1, you're missing 8. So this is just a diagram to show you that the fetus so this just show you the father is balanced because it's inverted 6, but the kid during pairing resulting in deletion or deletion and duplication in this patient. So the genomic structure, you could structural variation, you could have deletion, duplication, inversion, insertion, and translocation. But I don't have time to talk all of this, but just to show you one of the examples. And in the Baylor array design, why that ours is a little bit superior, because before 2004 we spent a decade just have the other gene discovery, and then after the gene discovery, when the array became available, at the beginning it was a bacteria artificial chromosome array, back array, and then later on we emulated into adjuvant array. And the reason for that is because we know that, for example, we know the Rett syndrome, we know at that time there was at least 40 syndromes that we know, and we put it into the array. So we using, because we know the gene localization, so we could put the probe into the right place so that we could detect them. And so this would illustrate to you a little bit about our design of the array. The design of array, this is what we call evolution of CMA. So we started in 2004, and we had 366 back containing 40 genome disorders. Remember I said that prior to 2000, we spent a decade just discovering different genes, and then immediately we put it into fish, using it for fish for detection of these syndromes. And then and when the back array come along, then we put them into back array back array. And then when the Agilent in 2007 come along, they had the 44K, and then they double into 105K, and then they have 180K, and then to be 400K. And then by the time of 400K, we were able to put more put more genes with exon coverage, and we find the genes from the omen, and we put them in. At that time, we would have almost close to 2,000 genes with exon coverage. So here is the glimpse of our 400K that we have. You can see this is an erection gene in our XN1 gene. And we will put the oligos into the exon region. Each exon will have 4.3 oligos, because depending on the availability of the probe. And then we have certain distance, like intronic resolutions, 1 probe for every 10 kb. And then between genes, then the baseline is every probe for 30 kb. And then also that we have the mitochondrial genome there, and then we have we had about 2,000 genes that with this exon coverage. And here to show you that the red dot is our array, and then you could see the Illumina array, and then you see the alpha array. The probe distribution randomly distributed. So like this Cohen gene, VPS13B, each exon, we have the gene there. Each exon, we have the probe there. And then, for example, the first one, we had the probe so we could detect them, where in the alpha or Illumina, they don't have the probe in there, so then they probably will miss it. Same as the Shank gene that, that we had to probe on the exon, showing you that the probe on the exon coverage. And here's the article that we published, that we had 2 patients. One is when we published 2 children associated with Rubinstein-Tabby syndrome, detected by a CGH. The first patient is the CREBB, P gene. And you can see below that the deletion, and then later I'll confirm that the deletion is from exon 27 and 22, 28. And then at the same time, we have a patient had a deletion of EP300. And you can see that the deletion on below, and the deletion is from 24 to 27 exons. So these are the 2 patients. Rubinstein-Tabby syndrome. On the right side, it's a typical Rubinstein-Tabby syndrome. The gestalt of the facial feature and the thick thumb. They usually associate with growth retardation, dysmorphic features, skeletal abnormalities, mental retardation, broad and angulated thumbs, and heliosis. But then they also find that EP300 had the similar features, but not so pronounced as the Rubinstein-Tabby syndrome. The EP300 is the phenotype is milder and lacing of a wide and angular thumb. At the time we published paper, there was only six of these EP3, 6 patients associated with EP300. And both genes act as a transcriptional co-activator. So since then, there's many many microdeletion, microduplication are reported. So this is conclude this section of the lecture. And this just for you to remember that, the evolution of cytogenetics and cytogenomics. First, we have the chromosome undented, and then we have the dented chromosome, and we have the fish, and then we have the sky, and then we have sky or race or CGH, and then we have the array CGH, CMA, and then we have the genome, whole genome sequencing.